Hello, everyone. We're about to get started. And I'm going to read the William and Mary's Land Acknowledgement and the William and Mary's Statement on Slavery and its Legacies. William and Mary acknowledges the indigenous peoples who are the original inhabitants of the lands our campus is on today. The Taranhakanadawe, Chickahominy, Eastern Chickahominy, Mattaponi, Monacan, Nansemond, Nottoway, Pamunkey, Potomac, Upper Mattaponi, and Rappahannock tribes, and pay our respect to their tribal members, past and present. The Board of Visitors acknowledges that William and Mary enslaved people, exploited them and their labor and perpetuated the legacies of racial discrimination. The board profoundly regrets these activities, apologizes for them, expresses its deep appreciation for the contributions made by the African-American members of its community to the vitality of William and Mary then, now, and for all time coming, and commits to continue our efforts to remedy the lingering effects of past injustices. Good evening, everyone. I'm Dr. Juwan Johnson, and I'd like to welcome you to the final segment of our Sankofa Genealogy Workshop series. Through this series, we've had the wonderful opportunity to hear from some great genealogists and family historians. The series was purposed to provide beginners and advanced level genealogists and family history researchers with the tools to delve deeper in family and community histories. And if you'd like to check out those previous sessions with Ms. Nika Sewell-Smith and Ms. Besida Carthon-White, you can visit our Lemon Project YouTube channel, which I believe will also be in the chat, and check out those wonderful, wonderful workshops. Also, if you haven't visited the Lemon Project blog, I encourage you to do so. We have some fascinating stories there. With our staff of researchers have presented some very compelling topics that I encourage you to check out. We'll be releasing more blog articles this coming fall. Tonight's workshop, which I'm very excited about, is titled "We Carry the DNA of Our Ancestors, a Genetics and Genealogy Workshop with Dr. Raquel Fleskes." Dr. Fleskes and I had the pleasure of meeting at two of the community convenings related to the Nassau Street First Baptist Archaeological Site. And we've exchanged and had some wonderful conversations, and I'm glad that she's able to join us. She is an anthropological geneticist investigating the population histories of colonial North America using ancient DNA. She is also a postdoctoral fellow in the Department of Anthropology at the University of Connecticut. In 2001, she earned a PhD from the University of Pennsylvania after being awarded a dissertation completion fellowship for the 2021 academic year. Previously, Dr. Fleskes received the Graduate Research Fellowship at the Wolf Humanities Center. From 2017 to 2018, she was a visiting researcher at the University of Tennessee's Ancient DNA Laboratory. Without further ado, let's give a virtual applause to welcome our special guest speaker, Dr. Raquel Fleskes.
Thank you so much for that kind welcome. I'm so excited to be here today. Dr. Johnson and I have talked a little bit about our overlaps with genetic research and genealogical research, and how we both feel that genetic research and genealogical research really kind of work hand in hand. So I'm really excited to be here to talk to you guys a little bit about genetic genealogy, about how we can be critical researchers when we get genetic testing results, and hopefully for you guys to take some tools away with you when you're doing your own family history research. So I'm going to share my screen, so it may take just a little bit for me to get set up, so bear with me. Okay, let's see. All right, you guys see my screen all right? Okay, great. Okay, so I've titled this presentation, "We Carry the DNA of Our Ancestors," and I really love this title because in my work down in South Carolina on an 18th century African burial ground site called the Ancestry African Burial Ground Project, where we have done DNA of the ancestors that were found there, one of the community members came up to me and says, do I share the DNA that is found within of these ancestors? Am I a descendant or am I related? And we had a conversation about how DNA can also embody all of these generations of people that come before us, right? And we get the honor of being able to study that when we study our DNA. So that's kind of the motivation behind this title. So I guess we'll just try to get started. I'm going to try to stay within our 45-minute timeline. Okay. So starting here with genealogy, right? Genealogy, in genetic genealogy, I like to think of as the backbone, right? It is the scaffold for which we form our questions. We think about who we are, where we come from, what our family history is. Genealogy, we focus on building family trees, right? So in this infographic here, we have this one individual in the middle, and he's built this family tree of relationships according to himself. So he has his brothers, his mothers, his grandfathers, and great-grandfathers, right? Now, in order to construct these family trees, we oftentimes can use things like oral histories or stories that we get from our parents. We call our mom and ask her, hey, tell me a little bit about my grandmother, or tell me a little bit about my great-grandmother. And maybe some of you in the audience are these family keepers, right? Keepers of the word, keeper of these family histories. And I like to think that this is what really brings life into the work in genealogy, right? The stories, the little questions and the personal vignettes that we can collect. So if you're able to record these oral histories, please do, because these are really the jewels and the treasures. Now, when oral history sources maybe pass through time or we don't have access to them anymore, then we are left with documents and archival research as kind of the framework through which we can view the past. So we're looking at documents for things like names, birth dates, death dates, locations, relationships, right? Things that are going to give us clues as to who this person was in relationship to yourself or to another family member, and who they were in their life. What are their stories? Now, sometimes in genealogy, we can hit roadblocks, right? So I always like to tell the story with my personal family history. On my mother's side, my, I think, great-grandmother was an immigrant from Austria-Hungary at the beginning of World War II, and fled during when Nazi occupation was beginning to start, came through Ellis Island, but we are unable really to move really past this point because we're unable to locate her records, because we think likely her name was changed during the adoption process, and thus it's kind of a little bit of a wall for us. Now, there are other circumstances like slavery, where names of persons that are forcibly transported are recorded incorrectly, or names are changed. And this can be a bit of a block, right? Or in conditions of enslavement, oftentimes names are not properly recorded. Demographic information is not preserved and with the same level of care than, let's say, the white male landowning individuals, right? And when we get deeper in time, we often come across things like not all documents are preserved. We have things like fires, courthouse fires that will, destroy lots and lots and lots of documents. And also histories that are often unrecorded, histories of women, histories of folks who are enslaved or indentured servants that we just don't get the same granularity. Now, I know that in other sessions in this research series have talked about how we can circumvent some of these roadblocks, right? So this is not the end, right? We just might want to think about different strategies that we can go about to try to take a different route to see if we can get answers to these questions. So again, it's not the end, right? It's just maybe we need to take a different turn in the road. But when we are critical genealogists, we also have to be evaluating the sources that we're looking at, right? We have things like documents where ages are changed. My great-grandmother was guilty of this. She decided to make herself 10 years younger on the census. So we can see that gap when the census comes out. She wanted to be a little younger for her second marriage. So, but we also have things like fake death certificates or mistaken parentage or all these hidden family histories. right? That they did not necessarily want to be written down. So it's always very important to double check all of our sources, right? And to be critical researchers, because as genealogists, we are researchers, right? We are using skill sets of historians to evaluate documents, to piece together our own personal family histories. Now, to be a critical genealogist, I think also aligns with being a critical geneticist, right? So genetics can also be used to look at our family history, right? We can assess our autosomal DNA, our mitochondrial DNA, and our Y chromosome DNA. And we're going to talk about all three of those in this presentation to think about our ancestry or similarities with contemporary populations and relatedness. So how we relate to other folks genetically. So if I have a second cousin or an aunt and an uncle, my DNA will be able to tell me that. And over the last really 10 years, we've just had a growth in the amount of DTC or direct to consumer genetic testing companies. So these companies will send you either a vial, right, where you will either spit in and deposit saliva, or you will take a cotton swab and rub between your cheek to give you some buccal cells or cheek cells. And they'll take that and extract DNA and then sequence your DNA for specific markers that they are analyzing. And so we get different types of tests, the mitochondrial, Y chromosome, and autosomal, and they'll look at things like ancestry testing or relationship estimation. And this has really opened up a new platform for genetic genealogy because it's made personal genetic testing more accessible than it had in the last, really since the 2000s when the human genome was first published, right? So we can see here this just astounding growth in the number of samples based on these companies, Ancestry DNA, 23andMe, MyHeritage, etc. However, genetics can be complicated, right? It's a really high learning curve. And like being a critical genealogist, we have to be a critical geneticist. Oftentimes, because these methods are complicated, ancestry and relatedness inferences can often be overinterpreted. And this is largely because this is a commercial endeavor. These direct-to-consumer ancestry testing companies are trying to sell you a kit. They're trying to sell you connection, heritage, and really ancestry, right? So we need to be aware that sometimes the marketing strategies that are used may not be fully accurate or kind of maybe bend a little bit of the truth, or they're not going to tell you how things are a little complicated. And this can lead to concepts like genetic essentialism, or the idea that DNA is the ultimate arbitrator of truth, that there is no other gold standard but DNA. And that's really not the case. Genetic methods are a product of the methods that are used to produce them. So what I hope to do in this presentation is to go through some of these critical lessons of genetics to equip you with how to be a critical genetic genealogist. So when you get your genetic testing results, you can really think through what results you're getting and what they actually mean to avoid any kind of overinterpretation. So again, thinking about genetic genealogy, right? Again, genealogy is the scaffold, right? This is what we're building our questions about. We're using documentary research and oral histories to create these family histories. And I view genetics as an additional tool within this genealogical framework, right? It can be used to verify or identify biological relatives or relatedness, and also assess similarity with other populations, right? But to build a critical genetic genealogy is to really understand how to properly use and evaluate these genetic tools in genealogical research, right? So how do we make sure that we're using these tools correctly to answer the questions that we want to answer? Okay, so that's kind of where we're going to go for this presentation. Most of our presentation today is going to talk about the science behind direct-to-consumer genetic testing, and then we're going to kind of tie it all together at the end with some overarching conclusions and things to think about. So in this part, what we're going to really do is think about what a genetic ancestry test. There are kind of three main tests that people get done. And this is across all different platforms, Ancestry, DNA, or 23andMe. They usually offer an autosomal DNA test, right? This is your whole genome. They'll usually offer a Y chromosome test and a mitochondrial DNA test. And we're going to talk about what each of these three tests do and how it informs relatedness or connections between living people, and then also connections with ancient individuals, archaeological individuals, and then also how it's used to interpret ancestry or genomic similarity. All right, so our first segment that we're going to talk about is autosomal DNA. So autosomal DNA, will often hear it's also called whole genome DNA, genomic DNA, full genome DNA, nuclear DNA. These are all the same things. They all mean the same thing. So there's a lot of different words that get thrown around. So I just want to make sure everyone's clear on the verbiage that gets used. Okay, they all mean the same thing. We're looking at our total ancestry. Okay, so this is our nuclear DNA. That is our 23 chromosomes. We're going to talk about the structure in just a second, but really what we need to think about with nuclear DNA and autosomal DNA is that it contains the DNA of all of our ancestors, right? So your DNA comes from your mother and your father, your biological parents that get mixed up every generation. Their DNA comes from their parents, their DNA comes from their parents. And so this is how your genome can reflect your total ancestry. Now, our autosomal DNA is found in all of our cells and the nucleuses of our cells. So if we go back to biology 101, here's our little diagram of our cell, and we have our little nucleus in the middle, right? And so all of these little cartoon X's are our DNA, right? DNA is basically one long strand that gets very, very tightly coiled. So like, think about, you have a rubber band and you just keep twisting it and twisting it and twisting it, and then it'll start to kind of twist on top of itself because it's so highly twisted. That's what a chromosome is. It's basically just a highly concentrated and twisted segment of DNA, okay? So our DNA is divided into 22 autosomal chromosomes. So this means, right, 22 pairs of these little X's that we see here, plus an X and a Y chromosome that determine our genetic sex expression. Now our genome is huge, right? It's estimated to be around 3.2 billion base pairs. Now a base pair is each one of these little, I want to say little brackets that connect each of the backbones of these DNAs, right? So what we do when we do genomic sequencing is that we kind of get the order of these different DNA bases, right? And that's kind of what a DNA sequence is, okay? So the really cool thing about autosomal DNA is its inheritance pattern. And you're going to see throughout this talk how our DNA is inherited gives us clues into how we use it for ancestry testing and relatedness estimation. So as I said before, our DNA is passed to our offspring with approximately 50% coming from each parent, right? So the sperm and the egg come together and fertilize, and this is where you get both of the chromosomes coming together. So hypothetically, let's say this is let's say this is myself, right? And I have, this is our chromosome. So if we go back, that's this little X segment, but just in kind of cartoon form. So we've actually split it in half, because a chromosome is actually just a pair of two chromatids, right? So again, this is our chromosome, okay? And I get this little brown block from my mom right here. This is her brown block. And then a blue block from my dad over here, okay? And you can see if we go to my mom, then this generation, she gets her little brown block from her grandmother and this red block from her grandfather, okay? So we see 50%, okay? Now, however, if we look at this, then you see that we actually lose DNA though, right? We don't have any of this lavender reflected, that gets lost, or this purple, okay? So in reality, our inheritance is a little bit more complicated. We have recombination in our autosomal DNA. So when I say that we get approximately 50% of each parent, it kind of gets all mixed up. And the mixing up allows us to hold on to segments of our ancestors so they get passed down to our DNA. So for instance, now we have this blue and brown block, right? That blue and brown block, and you can see that's from the grandparents. This red one has stayed intact in this example, which can happen, but really you see this other chromosome or this other chromatid here is a combination of the blue, orange, and green that he gets from the grandparents before them. So this is really important to think about, how do we get our DNA from the ancestors that come before us? Now we can measure the similarities of our chromosomes using a concept called identical by descent, or IBD analysis. And basically this happens when we can measure how long a segment of a chromosome is. And if you share the same chromosomal segment, it's likely because you have a common ancestor. So for instance, in this example here, we have a little bit of an orange segment here. And if we think about, okay, well, where did this orange come from? Well, we have it here and we have it in this third generation. And this other individual also has an orange segment and it's coming from that same source. So we can measure how long this segment is because to determine how closely related somebody is. So this is a concept called centimorgan. So here we get with our dense science for the day. So a centimorgan is basically, think about it as a DNA ruler. Okay, so let's say our DNA, let's blow it up and let's put a ruler to it, and we're going to measure it not with centimeters, but with centimorgans. Okay, so the longer the centimorgan is, the bigger the chunk of DNA that you have in common. The shorter it is, the shorter the amount of DNA in common. Now, when we're thinking about what this means in terms of how closely related you are, we have to go back to the concept of recombination, right? Every generation, we get split up by 50%. So like, for instance, let's use this pen. If this is one centimorgan, right? And let's say this is what I got directly from my parents. After one generation, that's going to be divided in half. So our centimorgan is going to be half as long. And then again, it's going to be 50% of that. So over time, the segments are going to get shorter and shorter and shorter. So the shorter the segments are, the more distantly related you are. Okay, so that's kind of how we assess how closely related are we using the autosomes. Okay. So for instance, we can query somebody's DNA against another person in a database to determine, if you may be a cousin with them. So this is Roberta S. She runs this blog called DNA Explained, which is a great blog. And she shared that she has, used Ancestry DNA, which has this platform about shared centimorgans, and found out that she has a first or second cousin with this individual named Lonnie, where she shares 357 centimorgans, right? So that's the amount of DNA across 20 segments. So this is in different segments, right? So 5% of her DNA is shared with Lonnie. So this indicates that she is a first or second cousin, okay? So we can take this even farther, right? So the logic of 50% decay at every generation due to recombination can help us infer what degree relationship you have. 50% is first degree. 25% is a second degree relationship. So an aunt and a niece, or an aunt and yourself, or a niece and a nephew. And then we have third degree, all the way down to really about 8th degree, which is our third cousin once removed. Now, as you can see though, as we keep going down, the amount of shared overlapping DNA goes down, right, to a very, very small percentage. So this brings us to our critical lesson 1. We can actually only go back so far, greater than 12 generations, right? At some point, we go back and this amount of shared DNA is just too small. And so this brings us to, I think, a very important concept. So this graph here reflects our genetic ancestors. So let's say you are in this middle here, here's 50 and 50. So your parents' DNA, their parents' DNA, their parents' DNA. And as you see, every generation, the contribution gets smaller and smaller and smaller until it's but a fraction, right? And after that, the contribution of these non-genetic ancestors is so marginal that they actually don't contribute anything to your genome yourself. So your genetic ancestors are much smaller than our genealogical ancestors. So our genealogical ancestors are all of our ancestors that came before us, right? Whose lineages coalesce to make us today. but this means that they may not have contributed genetic data, right, to our genomes, okay? So we have way more genealogical ancestors than we do genetic ancestors. So here's a graph that's showing the number of ancestors really past like our 8th generation just skyrockets. It exponentially proliferates, right? So this is our number of genealogical ancestors, really just shoots up. And then our genetic ancestors kind of plateau and stay the same, right? And then it kind of runs out. So the probability that somebody is our genetic ancestor is very, very high up until about the 6th or 7th generation. And then it really decreases because the amount of shared DNA overlap is so small. So this is our critical lesson too. We have way more genealogical ancestors than we do genetic ancestors. So that's how we infer relatedness using autosomal DNA. We are comparing chromosomal segment length between each other to determine, and the longer the segments that we have, the more closer we are related to each other, okay? Now we're gonna move into ancestry testing using autosomal DNA. So ancestry testing is basically when we are assessing similarity between you and another reference population, okay? So this is already a little bit confusing because when we think ancestry, we think ancestors, right? We think people who come before us, when really ancestry testing and how the concept is actually being methodologically executed is really similarity. Okay, we're comparing your DNA with those found in reference populations across the globe to see how much DNA or how many markers in your DNA do you share in common. So that's one thing to keep in mind already, okay? So we oftentimes will see testing companies have things like you get a lot of ancestry percentages, right? I think this is maybe Ancestry DNA, right? So this person has 48% DNA from southern and eastern India, and then 15% from Senegal. And Francine's over here, she has 16% from Eastern Europe, right? So we are very used to seeing these percentages, right? These pie charts that will plot your ancestry in a very 100% complete circle to say, this is where you're from. Now, the reason why we can assess similarity, right, not ancestry, but similarity, is because of our shared migration histories, right? So let's take it back to about, 100,000 years ago, or 200,000 years ago, all modern humans evolved in Africa, right? So for hundreds of thousands of years, we have anatomically modern humans moving and migrating and peopling in Africa, right? So we think about here's our map of Africa. And if we have all of these different colored dots, let's think about those as different colored marbles, right? Representing our genetic diversity, right? So each different marble is a different color. So how genes become more diverse is just by time. So I like to think about it as like a popcorn machine. So you like put your popcorn kernels in the microwave, you put it on for a couple minutes, and after a while we get one popcorn pop, another one pops, and another one pops, another one pops. And it's a byproduct of time, and it's random. We don't know what popcorn kernel is going to pop first, right? And it's the same thing with genetic diversity. This is a concept called genetic drift. So it's just a random accumulation of mutations in our genome that really don't do anything, right? But the more time a population is incubating, the more diversity they're going to have. So this is why African populations have the highest amount of genetic diversity comparison to all global human populations. And all global human populations are just a subset of this original genomic diversity in Africa. Okay, so, we're incubating in Africa, then we have a subsection of this population that leaves Africa, maybe, around 100,000 years ago, different, times that people come. It's not all in one pulse, there's multiple waves, but folks will leave Africa, they go into the Near East, then they spread into Europe and down into Indonesia. But you can see in these migrations, a subset of the genetic diversity is going with them, right? So we don't have every single color of marble in the Near East, right? And it's less. And then when we go into Europe, we lose the yellow marble, right? But again, they're all the subset of this original diversity pool. And so eventually what happens is that, we have populations that go into Australia and then across the Bering Strait into South America. So we have populations in South America that have yellow and green marbles that look different, a little bit different than, let's say, populations in Africa that have all these different colors of marbles, okay? So this is going to basically say that, when we're looking at genetic diversity, it's all connected, right? It's all a variant of itself. So the types of genetic diversity that we're looking at in ancestry testing is called single nucleotide polymorphisms, or SNPs. So we also call these markers to just keep it simple, right? But a single nucleotide polymorphism is one base pair change in the genome, okay? So like this individual in purple has an A base here, and then this individual in blue has a G base, and then this individual in orange has a T base. We would call that a SNP because it's different at that one position in our DNA base, okay? Now these SNPs randomly arise over time. Most of them don't have any effect. They just kind of are there hanging out in our genome, okay? They don't do anything. So also, when we're talking about genetic diversity, it's really important to talk about that most of this diversity is actually within the population itself. So most of our genetic variation is actually found within the same population. And that's because, again, thinking about that popcorn model, right? Those populations are kind of breeding with each other, and over time, right, we just get a more diverse population. So actually the variation between populations on the same continent and between different continents is actually just a very small subset. And also when we put this in context of our 3.2 billion base pairs, we're looking at like maybe 700,000 markers in our DNA. So this is a very, very, very small subset of variation within human populations. Humans are approximately 99.5% genetically similar. We are a highly similar species. So these genetic differences really are just, again, very, very small little blips in our genome that are a reflection of this human migration history. So, okay. So what we would do then is we compare the DNA that are found in different populations across the globe with your DNA, right? And we would look at these SNPs. So for instance, here is a map of human populations that are in this 1,000 Genome Reference Project, right? This is a very big endeavor where they sequenced a lot of genomes from across the globe. Now, what they've found is that if you look at these pie charts here, everything in gray are SNPs that are not unique. They're shared across the continent or with other populations, right? Things that are in yellow, or let's say in these kind of lighter shades, are ones that are private to the continent. So they're found, let's say in this example, in African populations, right? Or in this one, the light green is in Asian populations. And the darker shades are specific to the population. But so when we look across this map, the frequency of the dark colored pie chart is so small in comparison to the total number of SNPs that are found in each population. Okay, so this really segments down the SNPs that we're actually able to look at. Again, our population difference is really not that different at all. So this is then how they will determine your percentages. The percentages for these ancestry tests are based on the numbers of SNPs that are tested. They're not the reflection of your entire genome. So again, thinking about the 3.2 billion base pairs, our 22 autosomes, and our 6 chromosomes, when we get a percentage that says I'm 48% Irish, folks tend to think, well, that's my entire genome is from Ireland. Well, or 48% of it is from Ireland. And that's really not the case. It's 48% of the markers that they are studying, or 48% probability based on their reference populations. That is what your percentage means. So this percentage is a byproduct of the number of markers that are being tested, okay, first of all. So this is a plot from another genetic genealogy blog, where they've been able to access the number of SNPs between different ancestry testing sites. So you see Family Tree DNA has about 700,000 SNPs, or 700,000 markers. 23andMe has about 161. I think this is about 2018. I think this was put out. So if you take a family tree DNA test and then take a 23andMe test, they have only 14% overlap in the SNPs that are being tested. So you can take different genetic tests and get different results. And that's because they're using different SNPs. So this should be a flag for us when we're thinking about how rigorous are these tests, or what is this result really meaning? These results are a product of the test that is unique to the company themselves. Okay, it is not a definitive reflection of your ancestry or where you come from, right? And that is just the reality of what it is. And when we really think about science, right, as being a human produced endeavor, right, these tests then seem to make a little bit more sense, right? Now, in addition, what makes these ancestry tests also susceptible to the methods that are being used is that it requires very good representative reference populations to make the assessments on what SNPs, what markers are informative, which ones we're going to use to compare, right? If we don't have a good sample, we can't really make our guess of what SNPs are going to be good are going to be as good as the sample that they come from, the population that they come from. So if we don't have an accurate or representative reference population, our results are going to be a little off. And this is a really big problem for genetic ancestry testing. So for instance, the number of individuals in the Ancestry 2018 panel was 16,600 individuals that came from 43 populations. That is not a lot at all. 23andMe had 11,000 coming from 45. And we don't know what populations they are. We don't know how many individuals are in a population. We don't know what they're considering to be a population. We could have 10 individuals that are classified as Yoruba. And as we all know, Yoruba is an extremely diverse cultural and religious group that spans many different regions in Africa. So somebody who is of Yoruba in Nigeria may have a very different genetic profile than somebody who's from Yoruba from, let's say, the Gold Coast. So we have to be this is a big red flag for me. So, we have to think about, what are they basing all these results on? Representative reference populations are very, very important. Okay, now we're going to shift towards doing mitochondrial DNA. And we're going to talk about mitochondrial DNA and Y chromosomal DNA kind of together, because if we think about autosomal DNA as being our total ancestry, right? Our mitochondrial DNA and our Y chromosome are going to represent our maternal and paternal history. So we sometimes call them uniparental histories, okay? And we've gotten through all the hard stuff. The autosomal stuff is the hardest stuff. So if we got through that, then we can get through this easy peasy. So our mitochondrial DNA is also referred to as our mtDNA or our mito. Sometimes you'll hear mito thrown around. Again, they all mean the same thing, okay? They're referring to a very small plasmodial DNA. So you can see here, it's a little circular DNA, okay, that's independent of our genome. So our mitochondrial DNA is found in the mitochondria. This is a cellular organelle that's contained in all of our cells, right? So the mitochondrial DNA, because it's located in the mitochondria, has a different inheritance pattern. It is inherited only through the mother. And this makes it incredibly special because you get your DNA from your mom, her mitochondrial DNA, it comes from her mom, and her mitochondrial DNA comes from her mom. So it provides a straight, direct maternal snapshot into what that direct maternal history is, which is really, really awesome. And it will go back for thousands of years. So unlike autosomal DNA, where we, after about 12 generations, the signal from these ancestors really diminishes, mitochondrial DNA largely will stay the same. We might get a couple of extra mutations, but it still will be largely the same mitochondrial DNA. Okay, our Y chromosome is similar to our mitochondrial DNA, but also a little bit different. So We also hear Y chromosome is referred to as Y, or we'll hear NRY sometimes. NRY means recombining portion of our Y chromosome, to refer specifically to not the entire Y chromosome, but just the ones that are conserved. All right, because the conserved regions of our Y chromosome is what is of interest for us. So our Y chromosome determines our genetic male expression, right? Our sex expression. So if you are a genetic male, you will likely have one copy of your Y chromosome, right? And it's also paternally inherited, right? So this individual received his Y chromosome from his father, and his from his father, and so on and so forth. So like the mitochondrial DNA, it provides a direct snapshot into one line of our parentage, our paternal line, our father's history, right? It is paternally inherited. Now, what's different though about our Y chromosomal DNA is that it does undergo a little bit of recombination. So remember, recombination is when we switch 50-50 every generation, and that's how we reduce our segments. Now, the Y chromosome is a little different. It's a little bit small. You can see it here. This is our karyotype. So this is our X chromosome, and this is our Y. And the reason why it's so small is that every generation, it will tend to recombine with itself. So it does this little interesting recombination. And sometimes when you do that, you can lose segments of the Y chromosome. So this is why evolutionarily, we actually have a reduction in the Y chromosome over time. That's why it's so small. Okay, I'm going to bring my little screen up here a little bit. Oops. All right, so in looking both at our Y chromosome and our mitochondrial DNA, we talk about variations slightly different. We use a nomenclature system called haplogroups and haplotypes. So you might have heard of these before. These are the weird letter combinations that you'll see on the screen. You're like, what are all these Q's and R's doing? Well, this is basically a symbol for a motif of DNA mutations that belong to a certain group, okay? So a haplotype is a group of SNPs, a group of mutations, right? So these groups of these mutational motifs will form a larger haplogroup, okay? So for instance, let's say I have a mitochondrial DNA sample and it has a mutation at position 16311C. Now, this mutation isn't usually found in populations of Native North American descent, and we categorize it into a haplogroup called A2, okay? So what we do is we look for the distribution of haplogroups across the globe to match your haplogroup with other populations found. So, for instance, this is a map of the Y chromosome haplogroups. So we have Qs over here, which is a segment of your, the QM and the QZ is unique to populations in the Americas, and we have Ps that are unique to populations in Asia, right? And so P is our haplogroup, and then within P, there are going to be different motifs that form our haplotypes, okay? Now, if you see this map, it looks a little bit similar to our marble map, right? Of folks moving around across the globe. It reflects our migration history. But interestingly, Y chromosome is going to reflect our male migration history. So the map looks a little bit different, right, than on our autosomal map. Okay, so again, like I said, what we're going to do is basically compare the haplogroups that you have. They would sequence your mitochondrial DNA, sequence your Y chromosome, come up with those mutational motifs that are the haplotype, and classify you into a haplogroup. Then they're going to compare and see, where is this haplogroup found across the globe? So you can see the distribution. This is a map of mitochondrial DNA groups, and you can see we largely have African populations are largely composed of L haplogroups, and we have populations, let's say, in Europe that are lots of H's and I's and J's and U's, right? But we can see here, though, that even though we have regional distribution of haplogroups, populations are really different. Populations are not homogeneous entities, right? So, and haplogroups reflect this geographical migration, okay, instead of a specific population, okay? So for instance, you know, all, let's see, I'm trying to think, all Peruvian populations do not carry the same haplotypes, right? Peruvian populations have a very complex history of, right, of Spanish colonization. Later, we have a Chinese migration, and we have the Inca, you know, occupation, and very many cultures that came before them. And with them, they carried all of these different mitochondrial groups and Y chromosome groups. So really, we just have a large diversity that is present within each population. So you want to steer clear. This is our next critical lesson. Steer clear of tests when they say you have haplogroup L0A1 that belongs to the Igbo tribe. Well, not everybody in the Igbo tribe has the same L0A1, right? It's just maybe one person in their reference population has it. That does not mean that other populations also do not carry that haplogroup, okay? All right, so that's how we use our uniparental haplogroups, our mitochondrial DNA, and our Y chromosome to assess ancestry. Now we're going to talk about relatedness. So again, like we talked about, our mitochondrial DNA, for example, provides a direct maternal window into our direct maternal relatives, right? So this person's mitochondrial DNA will be the same as for great grandmothers, right? So hypothetically, when we share a haplogroup with somebody else, it can mean that we are directly related, okay? So it can suggest direct shared genetic and genealogical history. However, it does not necessarily guarantee that. And that's because a lot of people can share the same haplogroup. So if we think about when these haplogroups arose, they can different periods in time. For instance, K1B2B, this is a mitochondrial DNA haplogroup, arose approximately 5,000 years ago. So after 5,000 years ago, if we think about how populations expand over time exponentially, a lot of people might have K1B2B. So that means that you can have thousands of other ancestors or people that are connected to you, but maybe you're connected about 4,000 years ago instead of 5 generations ago, okay? A lot of people have the same haplogroup. So this is another critical lesson. Shared haplogroups do not necessarily mean direct relatedness. And this is where the genealogy becomes so important, because if we have the scaffold of genealogy, then when we do this genetic testing, we're going to be able to rule if it's a direct ancestor or if this may be more of a distant ancestor. Okay, and that's because many people share the same genealogical ancestors, right? If we think about how many ancestors we have, it's crazy. So if we start here at our 1st generation and go all the way up to our 8th generation, if we think about it, doubles, right? Almost doubles every time. So 8 generations back, we can have 256 ancestors, possible ancestors, right? If we keep going, then 256 doubles, right? And so we can have thousands of ancestors just 300 years ago, okay? Just 400 years ago. But however, like we talked about before, not all these individuals may be genetic ancestors, right? So again, we can share some sort of shared history, but they may not be reflected in our genome. So this then brings us to ancient DNA assessments, right? So ancient DNA is the field that I am in. So it involves extracting and sequencing DNA from archaeological ancestors to try to understand their population histories, how they relate to other people within burial grounds, how they relate to populations today. And so there have been recently different genetic testing companies that will compare your DNA to those of archaeological ancestors. And they'll say things like, are you related to the Vikings or ancient Egypt, King Tut, you know, tribes of Israel? Are you a Paleo-Indian? And I could go on about why these things are very highly problematic, but we'll focus on the science. There are a lot of issues with this, and that's because, again, like we said, many people can share the same mitochondrial or Y chromosome haplogroup. Just because you have the same DNA, let's say as King Tut, doesn't mean that you are the only person that has that same mitochondrial DNA, and that doesn't necessarily mean you're a direct genealogical ancestor. You guys could share an ancestor before King Tut was born, right? And be connected a little bit distantly related to them. Okay, so again, thinking about uniparental haplotypes can be a little bit deceiving because it makes you think that we are direct ancestors, when really it's more kind of can be a little bit more of a roundabout branch at times. Now, they'll also do things like assess your autosomal DNA and compare that to those of the archaeological ancestors. And again, this is something from, I think, genomelink.com, and they'll say you're 25% first farmers or 10% Indigenous American, which I don't even know what these groups are. A little bit problematic. But if we actually look at the science, what they're doing is assessing genomic similarity between your DNA and these DNAs from these ancient DNA samples. And they're saying you share, from based on their test, 25% of the same SNPs that are found in this individual. That is not to say that those are the SNPs that are only found in that population. You know, this could just be a European signature at this first farmers that you have. And if we actually think about it logically, you do not actually share ancestry, any autosomal ancestry, with individuals approximately greater than 200 or 300 years old. So it's actually not possible for you to share these SNPs that they're looking at, because they would have, the genomic signal would have been washed away. Okay, so, you know, we have to keep that in mind, that this is this is pseudoscience. Okay, and for individuals that are even less than 250 years, or possibly could share a little bit of genomic contribution, there's a problem of poor preservation, okay, for ancient DNA. Ancient DNA is just code for really bad DNA. It's like as soon as somebody passes away, their DNA and the organic material begins to degrade, and this degrades the DNA sequence. So for instance, here in this graph, this is a graph of the amount of proportion of DNA that they're able to extract. You can see from different parts of the skeleton, right? We have teeth in green. Petrous is a bone that surrounds our inner ear that's really dense and great for DNA preservation. But even that, we're getting on average around 60%, and with teeth, hardly anything, right? This is 60% of the DNA that they estimate to be present. So it's really, you know, we're really hindered by not having good DNA and ancient DNA context to do these really great autosomal relatedness comparisons that we can do, let's say, with living relatives. So this again brings to connection, having a genetic genealogical perspective, right? Genealogical research is so critical to help support the connections between living and ancient individuals. It will help verify things that maybe we find using the DNA, like let's say we do have shared haplogroups. Is this because of a more distant relative or because of a shared direct relative? Genealogy will help us answer that question. It will also help us confirm if autosomal relatedness is real or really just due to damage. That is often the case in ancient DNA. So for instance, in Williamsburg at the Nassau Street Project, we're going to try to extract DNA from the individuals that are found there. And because they are a little bit more recent in time, there is a chance that we might be able to do that. However, it's going to be really critical to try to do some genealogy if we can, to really verify the result that we find. If we do find haplogroups that are the same, or if we are able to get good autosomal data and try to think and look about this autosomal relatedness, if this is actually real or just an artifact. All right, so now we're going to kind of tie it up. I'm a little bit over time, so we'll kind of move forward. So in conclusion, our genetic genealogy can inform genetic connections to your relatives, genetic connections to your ancestors potentially, right? And genetic similarities to other populations. However, it's really important to think about these critical lessons along the way when you get your genetic genealogy results. Thinking about our genetic ancestors only go back about 12 generations. We have way more genealogical ancestors than we do genetic ancestors. Ancestry percentages are based on the numbers of markers tested, not your total genome. You can get different results from different companies. And this depends on the types of markers that are used and the types of reference populations that are being used. And populations are not homogeneous. They're not the same. Haplogroups and people move and change, and they're not just one haplogroup does not mean one population. And shared haplogroups do not necessarily mean direct relatedness. So I think if you keep these things in mind, it will help you think more critically about when we get these results, we can be a responsible consumer. Because again, at the end of the day, these direct-to-consumer ancestry testing companies are trying to sell you a product. They're trying to sell you identity, community, and history. And these tools can be so helpful and so important, but we have to take them with a grain of salt. We cannot literally interpret them. We have to kind of take them more broadly and more generally, because they are a byproduct of the methods that are used to produce them. Your identity is your identity. DNA is only one means of knowing about yourself and your history. So I'm going to end it there, and I'd be happy to answer any questions. And here's my contact information and a lot of great blogs. Genetic Genealogy is all about the blogs, man. They got great resources. So these are some really good folks that I've met that do good work. And I just want to say thank you to everybody for having me here today. It's been a pleasure, and I'm happy to answer any questions.
Awesome. Thank you so much, Dr. Fleskes. This is a fascinating presentation. Let's give her a virtual applause. So we're going to take about 10 minutes to answer questions. So if you have questions, feel free to list them in the chat. I see there are some already there, and we can respond to some of those. We'll give everyone a little time to get situated. We realize you just took in a lot of wonderful information. Okay, this one says, let me see. Yes, this video will be on the Lemon Project YouTube channel, perhaps early next week. Look out for that. You'll also receive a follow-up email from us with the link and some other materials that you may find useful. There's a question that says, there are so many people who claim to be a descendant of Pocahontas. Is this more about a shared haplotype?
Well, I think that's probably more of a genealogical question, right? Because that would require us having the haplotype of Pocahontas. And I don't think we've done any ancient DNA or found her remains or should do this type of analysis. So that I think is a byproduct of what types of documents we're looking at, our oral histories, the stories that are passed down to families. But also thinking about, we can have a lot of descendants, right? So, I don't know how many generations is between Pocahontas and today, but, you can, have quite a few number. But to verify that you are a direct descendant, again, goes back to genealogy, right? To going back to, building that family tree with the documents and the oral histories.
Correct. And this one is there a difference between DNA and DEA?
DEA, I haven't heard of that before.
I haven't heard of that one either.
Other than Drug Enforcement Agency, but I don't think that has to do with this.
So it says, is it misleading that the autosomal test result is 100% because people just assume the 100% reflects their genome? What is the best way to explain that 100% is not the sum of the tester? That's
a great point. So the 100% represents the sum of their analysis, right? Not the sum of your total genome, because they're not sequencing your entire genome. They're sequencing only parts that they're looking at for those specific variants. So that 100% is basically the SNPs that are being tested and kind of their own method of ancestry estimation that at the end of the day has to add up to 100. Now, what I wish that they would do is put like disclaimers like they do sometimes with food products that say, 100% is not your total ancestry. This is based on X number of SNPs from this many reference populations. Then I think people would be able to make that conclusion that it's not 100% of your own data a little bit easier than having to dig through all of the FAQs on the websites.
This one is, do you think 23andMe has a more complete DNA sequence form?
Yeah, 23andMe is great. I mean, so I know I'm a little bit of a critical scholar against these companies, right? But 23andMe, they do their estimations are still very good, right? And they do offer different types of tests. They do a lot of medical phenotype testing, so things like disease risk that a lot of other companies don't do. I think they have a whole genome option. I'm not sure. I know they're also looking towards a whole Y chromosome sequencing option, where you would send your DNA sample and they would give you your entire genomic sequence. But again, I'm not an expert, on specifically on all their different types of tests, but I think I think folks are moving towards doing a whole genome assessment.
And this was a comment from Besida White. It says, it seems like the real benefit of DNA testing to genealogists is the possibility of identifying unknown relatives on the basis of shared DNA.
Yes, that's 100% correct. And that's my biggest takeaway from, doing this work is that, if you participate, let's say in Ancestry DNA and do that shared Centimorgan project or go on GEDmatch, you're able to kind of approach your family tree from a completely different perspective, right? Whereas before, maybe you're building from your connections and going out, and it's a long process of tracing each line and getting each document, right? Well, your DNA is kind of going to do almost like a blast, right, against people who are in that database that you may share chromosomal segments with. And this might give you hints or clues that then you can try to maybe verify with genealogy. It might give you just maybe a different direction to go in with your genealogical research. Now, the only thing that I say about the chromosomal segments is that when you get very small, the probability that you're going to have a ton of third and fourth cousins goes like through the roof, right? So the smaller the segment of DNA, the less precise it is and more prone to maybe error that you have. So things like fourth cousins and fifth cousins, fifth generation, fifth degrees of relatedness may be a little bit more difficult to the probability of error is a little bit higher. But certainly between the first and third degree relationships, those are pretty solid and easy to rely upon.
This one is, do you know about the Hebrew E1B1A haplogroup?
Yes. So E1B1A1 is a haplogroup that is, so it's a Y chromosome haplogroup that is associated with Jewish populations and the priest populations. And so there was, and it's also been found in African populations that are Jewish, of Jewish religion. So there's a thought, or the rhetoric in the literature was that this was the priest haplogroup type. So persons who were of the Jewish priesthood or were rabbis had this E1B1A haplotype. Now, what we have found is that of course, not every population is the same, right? And E1B1A is a little bit, it's more widely distributed than to lineages of Jewish rabbis. Now, however, when we do find E1B1A in African populations of Jewish descent, it also has been used to interpret, lores about, Zion and, another, and has been used a lot for things like identity creation, nationalism in Jewish populations. So again, it's when we associate a haplotype to a specific cultural group, to a specific religious group, this is where, it may be associated with it, but it does not indicate that if you have E1B1A, you are supposed to be a Jewish rabbi or come from a lineage of Jewish rabbis. Many people will come from this type of lineage.
This last question says, how does the Y genome pertain to a woman's ancestry? Is it just a passing it on kind of thing?
So if you are a female, you do not have a Y chromosome. And sometimes that can be very frustrating for genetic genealogical research because you're like, dang it, I can't assess any of my paternal history. And that's just because we don't have one. So an alternative for this, if you are interested in investigating your paternal history using a Y chromosome, is to get a brother, get your dad, get an uncle, or somebody who is male, a genetic male in your family, and maybe encourage them to maybe go get their genetic test done. And so that way you can figure out what Y chromosome lineages are in your family. So, or if you have any offspring, have any children, they would inherit your male partner's Y chromosome. So yours kind of just stops as a woman, unfortunately. You don't get to pass it on.
Well, this has been great. Thank you all so much for joining us. Again, thank you, Dr. Fleskes, for being a part of this continued conversation. It doesn't end here. We will have the presentations available on our YouTube channel early next week. Also, we'll have more programs that will come forth in the fall. So keep a lookout on our social media platforms for the Lemon Project, and stay in touch with us. Thank you so much.
Thank you. And feel free to send me an email if you have any questions.
And there's Dr. Fleskes' contact information. Feel free to reach out to her, and have a wonderful evening and a safe weekend.